Okay, so we have discussed so far the introduction to cytogenetic method as a method to studying genetic diseases. Um, in this, we have discussed a brief overview of what the cytogenetics is, what the science basically deals about, giving you a brief, recap of what we have discussed. So far, it understands the the maturation or the phases, different phases of maturity of cells. We study the chromosomal abnormalities depending on the characteristic features of your chromosomes, analyzing those chromosomes using the method of karyotyping. Yeah, I have introduced you to the method of karyotyping. Then we discussed about the evolutionary background of it, the principle of cytogenetics, at what level do we study chromosomes, and for the understanding of karyotyping or banding techniques, we have specific phase of division wherein we arrest the cell and study the phase of maturation for that cell at particular stage of division. So that we have discussed so far. What are chromosomal abnormalities we have discussed, how to detect chromosomal abnormalities, and today we'll be starting with the techniques of cytogenetic method. We will not be in depth going into these techniques, but I will be taking you through the introductory and the application and the the technique part of it, but not in detail. Just to let you know wherein where all you can use these techniques, at what given circumstance can these techniques be used, and how do you use it, right? So these will be the basic area of discussion for today. So we'll be starting with the cytogenetic analysis. These are the techniques. I think I have already shown you what are the different techniques, the list of these techniques. Today we'll be starting with karyotyping, the first technique. So today this session particularly is talking about karyotyping and the fish technique. Okay, so starting with karyotyping. Now, karyotyping is one of the many techniques which help us to study the human genes or the cell behavior or the chromosomal abnormalities, and thereby studying the genetic diseases. So this term or this technique of karyotyping comes from the word karyotype, wherein karyotype is indicative of a complete profile of an individual's chromosomal setup. Any changes or any abnormality, any aberrations, the technical term that we refer to the changes in these arrangement of karyotypes, any deviations from the normal karyotype helps us identify what could be the probable genetic abnormality or a genetic disorder that a individual possess. So therefore, the process of karyotyping, in simple terms, if we have to say, it is a very close or a very detailed study of these chromosomes. These basically show the details of single chromosomes that we have, as in details with respect to their shape, their orientation, their structure, their size, their number, all forms of chromosomes and their details. Now we have discussed about chromosomal abnormalities, we have discussed about numerical and the structural abnormalities. So how you are already aware of what are different forms of these abnormalities. If there's any confusion, just refer back to that session again. But how do these these aberrations are visible is through the pattern of karyotyping, and there are therefore other techniques like fish, wherein you use the stains or the dyes to get that probes being hybridized with the specific target area of your chromosome of your genes, and then you observe it under the fluorescent microscopes and things like that. But karyotype basically is a full-fledged depiction of your chromosomes, set of your chromosomes, arranged with respect to their size and things like that, and therefore you are clearly able to see on wherein the shape of the chromosome is deviated, distorted, or the number of chromosome is less or more, So that's how you identify the changes in the chromosomal patterns. For instance, if I have to take this technique of karyotyping helps us identify and determine the sex of an unborn child. Now, when the doctors study a human karyotype, they look for some significant features. For instance, first they will see whether there are the desirable number of chromosomes, like we have 46 chromosomes present, right? So we first check, we confirm whether there are appropriate number present. Then what we'll see, whether there is presence of 2 identical chromosomes and then 2 sex chromosomes present. Then we'll see whether there is a distortion in terms of their arrangement or in terms of their number. So that is how we identify the deviations, if at all it's visible or not. Now question is, therefore, how is it performed? What is the technique of karyotyping? So as such, there is no specific requirement, specific protocol for doing a karyotyping. Like, you do not have to undergo fasting or any testing procedures are not there. What basically is done, the patient or a person who wants to get some conformity, so they come to the doctor or a counselor, and then they are recommended for their genetic analysis and karyotyping, therefore, if they are recommended as a technique. So what they do basically, a small sample of your blood, or depending on what is the target person. So if that is an adult person, or if that's a fetus, or if that's a newborn child, so depending on what kind of target person is, the sample of blood or of your bone marrow or of your amniotic fluid and placental tissue, depending on the level of the person, where the person or what's the target area, so that sample is collected. Then that sample is for instance, the blood sample is drawn from wherever, if you're talking about the fetus, that sample is drawn from that fetus. Now, different methods we have already talked about. Amniocentesis we have talked, Cronus villi sampling we have talked. So depending on what is suitable to that person, the technique is performed. Now, for example, if you're talking about amniotic fluid, you do the technique of amniocentesis. So that becomes very much appropriate technique for that particular test to be conducted for conformity, whether to conduct a prenatal diagnosis, to do the screening beforehand, you do the amniocentesis. Now what happens is, you collect the sample, then sample is basically placed on a tray. It is fixed using the chemicals like methanol and acetic acid in the ratio of 3:1, and then they are allowed to grow in the confinement of the laboratory based settings, right? So now the culture is ready. The cells from that growing sample is then stained. Generally, the G banding or the chiasma bending technique is one of the most commonly used technique to identify these chromosomes. These will be studying in the banding techniques, which will be taken for the next session. Now, this chiasma banding is the most common technique used to do the identification whether the person is having a normal set of chromosomes or there's some distortion, some deviation from the normal pattern. So using that chiasma reagent, which is mixture of different dyes, DNA is stained, right? Now when DNA is stained, it is becoming more easy to understand or observe the distortion in the microscopic observations. So therefore, that stained sample is closely examined under the microscope to study what kind of arrangement that chromosome depicts. Is there some numerical distortion? If there is some structural distortion, then that becomes clearly visible under the microscope. For instance, if we have to talk about a sample of karyotype, just look at this diagram. I have taken this picture from internet sources. Now, when you see about the good example of chromosomal aberrations is Down syndrome, Klinefelter syndrome, Turner syndrome. So if you see about the Down syndrome pattern, at the chromosome number 21, you have an extra pair of chromosome. One extra chromosome is there. Therefore, it's called as trisomy, and that is a deviation from the normal pattern of chromosome. Similarly, here you see in the Turner syndrome, in the karyotype of a person with Turner syndrome, you will see that there is a missing X chromosome. Right? So from a pair of X chromosome, you have one missing homologous chromosome. So therefore, that is identified as Turner's syndrome. Now when we know that a normal human being is having 46 number of chromosomes, out of which 22 are the autosomes and one being the pair of 6 chromosomes. So now when there is a disharmony between this set, there's a genetic condition, there is a genetic disease, or a probability of a person having the symptoms of a genetic disease, be it early onset or the late onset. Now, too many chromosomes, missing chromosomes, or there could be a mixed up bits of chromosomes showing the presence of certain genetic problem. Now, the state of chromosomes helps us predict about any possible genetic disorder in an unborn child using the prenatal testing and the diagnosis. So chromosomes carry information that is being passed to the cell. Extra copies of the information, or the mixed information, or any missing information can therefore lead to a defect of the conveyance of that information, or there could be an abnormality. The defect could be clearly visible later on. So what are the conditions in which we can probably use this method of karyotyping is? So there are n number of methods. It's commonly used for identifying particularly the genetic diseases in the prenatal form, but if at all they want to have a decision making process later on, if a person is having series of abortion or series of risked pregnancies, so that could be one decision making process for which they can use the karyotyping, whether there is a genetic deformity in their ancestry line or things like that. So one good example is abortus material, like in case of people having previous history of abortions, and they want to check whether they have any chromosomal abnormality within their paternal lineage or parental lineage for that matter. So both the mother and the father are going to get their genetic their genes, their chromosomes being checked for the abnormalities. So both ways, for both of them, there could be a possibility that any of the ancestral person would be having a mutated gene, and therefore that has been passed on over the generations. So therefore, this is one condition wherein you can use a process of karyotyping. Second thing is at the level of fetus development, using the amniotic fluid, plus when you have an advanced maternal age, which is generally considered to be more than 35 years or so, and there are previous histories of abnormalities in their parental lineage, or there are having history of abortions, so then that amniotic fluid analysis could be done. Now, apart from that, when after the birth you have a child with a rare childhood sort of neurological disorder, this is called ataxia telangiectasia, TELENGIECTASIA. So that is considered to be very rare neurological disorder during childhood. Now it causes the degeneration in part of your brain, controlling your motor skills or your balance or your sensory organs, your speech. So overall, facets of your development. Now this basically manifests itself in the early decade of your life, within the first decade, from the birth till 10 years of your age. So therefore, when you have a child with certain symptoms and you do not have a name to that disorder, So you do the karyotyping, you do the analysis of your chromosomes, the pattern of your chromosomes, and then you identify whether that rare form of disorder is there or not. So in that case, again, we use the technique of karyotyping. In the fourth method, there is bone marrow chromosomes for people who are having very, situations like leukemia or hematologic disorder, blood related disorder. In that case, we use the samples from your bone marrow to do the karyotyping. Now, another method is chronic villi sampling. It's also a method used during the prenatal diagnosis to detect whether there is any genetic or chromosomal abnormality of a baby within 10 to 12 weeks of your pregnancy, right? Because if you remember, in prenatal diagnosis, we have talked that there are certain limitations to these techniques. They have to be performed within a specific time period. If at all you miss the time period, then it would be dangerous for both the persons. So therefore, 10 to 12 weeks, or generally that phase, is considered to be more secure. You can perform those tests to confirm that if there's any genetic disorder or not at the level of fetus, right? So apart from that, there are different techniques like cord blood placental to determine the health or the developing conditions of the fetus. You use the placental sample to conduct the testing of the health or any other condition associated with that unborn fetus, right? So there are different conditions wherein you can apply the technique of karyotyping to study the chromosomal behavior, to study the chromosomal abnormalities, if at all they are visible or not, right? Moving on to the second technique for today, it is a FISH. So FISH as in fluorescence in-situ hybridization. Now it's a very powerful tool that we use for the detection of certain chromosomal abnormalities which are not being detected or not able to be detected by other conventional methods, for instance karyotyping. Okay. It is also identified under the category of molecular cytogenic method because its ability to identify a specific gene or a molecular region with direct visualization of the cells or the chromosomes under the microscope, right? Here the principle of this technique is based on the whole idea of hybridization, wherein we have a single stranded DNA. We are trying to hybridize it with a prop DNA, or say a complementary sequence of the target region of the DNA that we are targeting for study, to form a double stranded DNA. Then we use the dye to highlight that particular hybridized region, and then we study it under the fluorescent microscope. So that is the technique. So here, if you can see this diagram, here we are taking the gene, which is the target area from our DNA. We are taking that gene, we are cutting it at the specific area, then we are collecting the sample. So this is what the single stranded DNA looks like, right? Now we know that this particular region from DNA has to be studied. Similar or almost similar, or the complementary sequences of this pattern are fixed on the slide. That becomes the probe sample. Now here they are again denatured, the single strands are separated. Now these both are here, made to be hybridized by the process of temperature and the things like that. So now when you get the target sample here, there's a dye that we put in so that this region is specifically highlighted. Okay, we use the dye, and then this is being observed under the epifluorescent microscopy, or any fluorescent microscope for that matter, right? So if we have to talk about the techniques, The DNA probes, or the short sequences of single stranded DNA, they are tagged with a fluorescent dye, and then they are applied to the cell preparations on a slide for observations under specific conditions, as in temperature and things like that, the laboratory settings properly, so that it allows the probe to attach itself to the complementary sequence of the specimen that we are targeting to be studied, right? Then this hybridized section, or the hybridized region of DNA, is then visualized under the fluorescence microscope. Therefore, this becomes a very powerful technique to detect whether there are chromosomal abnormalities, such as any loss of chromosomal regions or the whole chromosomes, or say extra pair of trisomies. All of them can be very easily detected and studied. Now, this technique also becomes very important in identification of any genetic aberrations, like say this gene that we are referring to, ABL-BCR gene, is in reference to the breast cancer gene. So these aberrations, they're monitoring their progression, if it's tumor-like situation and it's seriously progressing or not, so that that monitoring could be easily done using these techniques. Now, with the advancement of technology, this whole genome can be screened continuously through, there's one more technique called CGH, which is comparative genome hybridization. In this test also, we have a test, we do the same method. It's also completely hybridized during the metaphase stage of the chromosomes, but it is only possible to detect the loss or the gain of a specific length of chromosomes. So therefore, we'll be talking this method in detail separately, but just to say that because of more advancement, we are also progressing with the whole genome being hybridized. So that is the technique of CGH, comparative genome hybridization. In this, we have a specific region only that we can highlight, because that probe has a fluorescent dye attached to it, and therefore that gets clearly visible. Only that region becomes clearly visible under the microscope after the hybridization happens. Okay. Now, what are the applications of this technique? So when you have any child with any form of development disability in family, or you have a history of it, so that becomes very easy for a parent to get tested what kind of abnormality they have, and then whether they should proceed on for next case or not. So that helps in a way as a decision making process. Right. Then it is used to diagnose, they evaluate the prognosis, and then therefore tailor a specific method of treatment for a specific disease that could be possibly there. So for example, cancer situations. So therefore a specific, a very contextual treatment could be tailored based on what exactly is the diagnosis or the prognosis of it. Then it is also used in clinical studies. For example, it helps in identifying the pathogens associated with the samples that we're getting. Then it also detects the presence of suspicious pathogens on the sample of patients. It also helps compare the genomes of two biological species to understand their evolutionary reactions, or the evolutionary relations, right? Then also used in, apart from the medical genetics, if we say, it also studies the microbial ecology part of it, and the interaction between the broader ecosystem, so to say. So this is about fish and the karyotyping techniques. Yeah. Okay. So if you have any questions, if you have any doubts, just let me know. Okay. Thank you.